Okay, hi everybody. There's quite a few announcements for today. Um, so first of all, this week is obviously most likely disrupted because of the bus strike happening on Wednesday. So a few things have been changed a little bit in response to that. So the first one of those is that I'm going to have office hours tomorrow in the morning rather than on Wednesday afternoon, in case people aren't going to be around on Wednesday. Wednesday's lecture is the last real lecture. It will go on as normal. It's sort of a light lecture. It's on quote unquote the future of neuroscience. And so there isn't going to be a ton of like really intense material that's going to be on the final, but there is a potential that small things could be on the final. So if you can't make it, you should still watch that lecture. Friday will be a review. I will be here to answer questions. However, knowing that many people may not be able to get here, I will also hold a review session on Monday, probably from 1 to 3. There shouldn't be any exams that day, so hopefully it won't conflict with anything. I have to find a room for that, so I'm working on that now. And hopefully the buses will be back to normal on Monday. I just posted this morning clicker grades for the terms. Basically, you have 2 grades out of 5, one for accuracy, one for participation. Participation is based on the number of sessions that you participated in. There were 16 overall, so to get 100% on, or get 5 out of 5, you needed to participate in 13 out of the 16. And basically, if you were less than that, then your participation grade is graded out of 12.8, because that's 80% of 16, which is the threshold for getting all the marks. The accuracy score is graded out of 26, because that was the highest score in the class. So the highest person gets 100, and then everybody gets a score relative to them. Right, and then finally, please take the time to fill out a course evaluation. These are really important. I do read them carefully. It helps me try to judge changes for the future. And so, yeah, I really appreciate it when people take the time to fill them out. I am fundamentally opposed to the idea of leaving class time for that, because I think it's like sort of silly because we have limited class time. We should do things that we actually benefit from all being in the same room. So I'm not going to give class time for it because I think it's better spent doing review and stuff like that. That's actually going to help you with the exam. However, having said that, that doesn't mean that it's not important. So please, like I said, take time to fill one out. Okay, that's it. Any questions about the logistical stuff? All right. So today we are going to talk about neurodegeneration, and we're going to focus on 2 diseases, Parkinson's and Alzheimer's. And I want to emphasize from the start that these are incredibly complex diseases and incredibly vast fields, that there's a lot known about both of these and a lot that's not known about both of these. And either one could be an entire course on their own. Right, so we're really just going to skim the surface and focus on a couple of things as we go that relate to material that we've learned about in other parts of the course. So Alzheimer's is the most common neurodegenerative disease, and this is an image just showing a slice through a normal brain on the left and a brain from a patient with advanced Alzheimer's. And you can just very clearly see the tremendous amount of neuron loss that's associated with this disease. And of course, the most sort of well-known manifestations of this at the earlier stages of the disease are cognitive impairment, memory loss, and that sort of thing. So Alzheimer's affects about 26 million people worldwide, and that's projected to increase a lot in part because of the aging demographic of the population. We don't really have, I couldn't find really good numbers for Canada, but presumably it's very similar to the US, where about 5 to 10% of people above the age of 65 are affected. And once you get above 85, the number jumps to almost half. Okay, so this is an age-associated disease. However, there are cases of early onset that probably have a different genetic cause, and we'll go into that, and where we can learn about sort of the molecular underpinnings of the disease. So the disease is characterized by dementia, as I mentioned, memory impairment, then basically breakdown of various systems, including language, spatial orientation, abstract thinking, and judgment. And the classic pathology associated with Alzheimer's are two aggregates in neurons or associated with neurons. One of them are these amyloid plaques, right? These are ones that you've probably heard of. They are made up of a protein called A beta that we're going to talk about, amyloid beta, that we're going to talk about in a little more depth. And these are actually extracellular. There are also these things called neurofibrillary tangles. These are made up of a protein called tau, which is a microtubule component, and basically, and these exist intracellularly. Okay, and these build up in various parts of the brain. So this image on the right is essentially showing areas there where plaques have been seen, and you can see that they're spread across the brain. They do tend to be very dense in areas like the hippocampus, which is associated with memory, right? But they are found throughout the brain. So one of the real controversies in Alzheimer's is the role of amyloid beta, and also, I guess, tau, but most notably amyloid beta, because it was the most prominently studied associated marker with the disease. And so, the initial thought and the way everybody kind of modeled how to tackle this disease was that there was some sort of insult that led to the production of these aggregates like the tau and A beta plaques, and then this led to disease. However, there is plenty of evidence, actually, that amyloid beta is not a primary driver of the disease. And so there is also the possibility that essentially an insult does something to create the disease, and then the formation of these plaques and tangles are actually a byproduct of the disease, right? So there are a cellular pathology associated with this disease, but not necessarily the cause of the disease. And if you look at the time course of plaques and neurofibrillary tangles building up in the brain of Alzheimer's patients, this is shown in these graphs. Okay, so basically this is time on the x-axis here, and during this period patients are cognitively normal. This MCI is mild cognitive impairment. Right, and then the final stage is full-on dementia. Okay, and so you can see that if you look at the buildup of A beta, it actually peaks and plateaus before there's any cognitive impairment on average. All right, whereas tau actually trails a little bit, and it increases before there's cognitive impairment, but it's still going up as disease progresses, right? And actually peaks then during the stages of dementia. So from a temporal standpoint, tau is actually more closely associated with disease symptoms than amyloid beta. And then the other graphs are essentially showing the progression of specific types of symptoms. So brain structure just refers to like neuron loss, essentially. And so you can see neuron loss is occurring here even before cognitive impairment, but then increases during the later stages. And then memory is shown in the purple line, loss of memory, and then clinical function in a general sense, right, is shown there. That obviously increases in the later stages. Any questions about this so far? Okay, so despite the kind of questionable role of A beta in disease onset, a lot has been studied about A beta, and there is evidence that A beta can at least sort of predispose the brain to developing Alzheimer's. Okay, maybe it predisposes the brain to the initiation of these neurofibrillary tangles, which are actually the primary driver of the disease. It's not really clear, but it does seem like there's a role. So A beta, amyloid beta, is actually a peptide product from a larger protein called APP, or amyloid precursor protein. Okay, so APP is a transmembrane protein that actually gets cleaved by a complex of proteins called gamma secretase, and under normal conditions actually does not produce amyloid beta. So it produces this other very similar peptide called P3 that does not aggregate into plaques. Okay, but something, whether it's a mutation in one of the components that I'm showing here, as we'll see in a second, or some other trigger, will tend to then, in some people, change the cleavage site of APP by gamma secretase and actually start producing amyloid beta. Okay, and then amyloid beta can go on to aggregate, and this is associated, as I said, with the disease. So as I mentioned, there are early onset cases of Alzheimer's, and so these are more classically inherited. So you can do genetics on these cases and see the inheritance of the condition in families. And so people have identified some major contributors to early onset Alzheimer's. And these include mutations in APP itself that presumably lead to overproduction of amyloid beta, or mutations in presenilin 1 or 2, which are both components of the gamma secretase complex. Okay, so again, this is evidence that amyloid beta is probably doing something related to the disease. Because these mutations are heavily associated with early onset forms of the disease. However, most patients are do not get Alzheimer's young. They get it when they're much older. And so late onset Alzheimer's disease also has a really strong genetic component. And the most famous of the genes that have been associated with late onset Alzheimer's disease is a gene called ApoE. So we're going to talk a little bit about ApoE. It's estimated to account for about 50% of a person's risk for late onset Alzheimer's disease. It produces one of the apolipoproteins within the HDL complex, which is just like your good cholesterol. And it's been found to actually bind to A beta. So there are these three known or dominant, or no, I shouldn't say dominant, these three very prevalent alleles of ApoE called ApoE 2, 3, and 4. And if you have ApoE 1 or 2 copies of ApoE 4, you're actually 15 times more likely to develop Alzheimer's disease. If you have only 1 copy of ApoE 4, then you are about 4 times more likely. And the important thing here, the functional connection, is that this E4 allele has been shown to bind to A beta less strongly than the 2 and 3 alleles. Okay, and we're going to talk about that in a second. So if anybody has ever done like 23andMe or some genetic test, I personally haven't, but apparently ApoE is one of the alleles that you can check if you so desire, and so you can see whether you have this increased risk, which is philosophically questionable whether you would want to know that, because it's one of those things that you can't really change in any way. But anyway, that's a personal thing. Okay, so there's a learning objective here to describe the major genetic factor underlying AD susceptibility and give a mechanistic explanation. So we're going to talk about the mechanism now. But before that, any questions? Okay. So there have been many studies on ApoE and its function. I'm just going to show some data from one paper. In this first experiment, what they did was take a bunch of people that had different allelic combinations of ApoE and just measure the amount of A beta that they found in the cerebrospinal fluid of those patients. Okay, and so these are very strange graphs. So we're going to look at the numbers first, okay? So these are the different allele combinations that they looked at: E2 over E3, 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 E4, and E4 over E4. So these are the homozygotes, right? And you can see that the average amount of A beta in the CSF is actually lowest in those E4 over E4 patients. Okay, second lowest in the E3 over E4, and then there's more A beta in the CSF of these other patients. So how is this interpreted? The interpretation is that this lower amount of A beta in the CSF is actually because there's more A beta left in the brain. Okay, so if ApoE is involved in binding to A beta and then clearing it from the brain, then if you have the ApoE4 allele and it binds less strongly, it's less able to clear it from the brain, so you see less in the CSF and there's more still left in the brain. That's the interpretation. So if you look carefully at the y-axis of this graph, it's actually a graph of the percentage of individuals with A beta levels in the CSF below 500. Okay, so this is, the higher bars actually mean lower amounts of A beta in the CSF. So it's just a weird way of graphing it, but these match those numbers, right? Because the E4 over E4 has a higher number of patients that actually have a lower amount of A beta. This has also been studied in mouse models of Alzheimer's disease. So there is a classic mouse model that basically involves the overexpression of APP, and these mice develop symptoms, both the pathophysiology and the actual sort of cognitive symptoms of Alzheimer's. And if you have mice and engineer them with different actual human alleles of ApoE, you can see that, and then stain for A beta, you can see that the addition of the E2 allele actually does a lot of clearing of A beta from the brain because there's very little left. Whereas if you add the E4 allele, then you have very little clearance, and so there's much more A beta that builds up in the brain. Okay, and this is quantified in the hippocampus over on the right, where again, the E4 allele is much less able to reduce the levels of A beta in the brain of this Alzheimer's model. Any questions there? So like I said, this is an incredibly complex disease. We don't know what the causes are. I'm putting this slide up to basically make the point of like lots of stuff goes wrong during Alzheimer's disease, you know, including the activation of certain enzymes that can lead to the phosphorylation of tau and the production of these neurofibrillary tangles. And at the center of it all here is this like A beta that's aggregating. But, you know, don't remember all of these details. I'm just showing this again to essentially say that it's complicated and we don't really understand exactly what the driver of this disease is. One of the really curious things about Alzheimer's and the role of A beta and APP is that it was a long time, or it's been a long time, without a really convincing demonstration that APP and or A beta actually do anything good. Okay, so like, why would you have a gene that all it does is when something goes wrong, you get Alzheimer's, right? That doesn't make a lot of sense. So, you know, do APP and A beta actually have a positive endogenous role in normal individuals? So there is a documented role for APP. If you make a mouse lacking APP, they actually survive and they do okay, but there are sort of mild cognitive deficits, suggesting that it must be involved somehow in neuron function, though the exact role is not entirely clear. So, presumably this role, though, is via non-A beta producing pathways, and maybe A beta is just like some negative byproduct that can happen. A study came out a few years ago, though, suggesting that A beta could actually have a positive role in at least one scenario, which is actually the clearance of invading bacteria. Okay, so if you take a mouse and you inject its brain with salmonella, which is not the most physiological, normal thing to have happen, but if you do that, a wild-type mouse will die over the course of a few days. That's what's shown in this black line here, right? This is survival over time, so you see the survival going down until all of the mice are dead. But if you do the same thing to a mouse that's actually a model for Alzheimer's, this is the 5X FAD mouse. You don't have to worry about the details. The point is that these mice actually produce more A beta than normal, and so they get Alzheimer's. Or Alzheimer's-like pathology, I should say. But these mice actually live longer than wild type, suggesting that A beta could have a protective role in the scenario of infection. And so if you look at the clinical score based on this infection, you can see that it's worse in wild type than the FAD overexpressing mice. And if you measure the actual bacterial load in the brain, it is lower in these A beta overproducing mice. All right, so this suggests that maybe A beta is sufficient to at least help clearance of this bacteria, right? So is it actually necessary? They also tested the APP knockout, And it turns out that the APP knockout might die a little bit faster when you inject them with Salmonella, suggesting that maybe actually there is like a necessity here as well, that even in the wild type mouse, there is some effect of A beta or APP on this infection progression. Any questions about that experiment? So from a cellular point of view here, they also showed that in these mice that overproduced A beta, the A beta, which is stained in green here, was found to be actually associated in space with the Salmonella. All right, so you see all this Salmonella aggregating, and it's kind of surrounded by A beta, suggesting that maybe A beta is like, encapsulating the Salmonella or something like that, and somehow helping reduce disease burden. So it'd kind of be like well, I won't even say that. Okay. Any questions there? Any questions about Alzheimer's in general? Because we're going to move on to Parkinson's now. No? Okay, so Parkinson's, also a very common neurodegenerative disease, also age-associated, right? So this affects about 1% of the population over 65. Much like Alzheimer's disease, most patients are late onset and sporadic, right? But there are early onset cases that have led to important discoveries about the mechanisms or at least the genetic involvement in Parkinson's onset. So we've talked a little bit about Parkinson's symptoms in the context of action selection, motor initiation, things like that, right? And of course, Parkinson's is associated with the loss of dopaminergic neurons. Some of the notable symptoms include bradykinesia, which is the slowness of movement, right? Which you could think of as actually predominantly the inability to initiate new movements because of these defects in the dopamine and its association with the basal ganglia. There is also a notable resting tremor, right? So that just means basically if the patient is holding their hand, let's say trying to hold it still or resting it, there's like this shaking, right? So this should not be confounded with either the slowness of movement, right? So some people get tripped up by the fact that, this is a movement disorder, like you have less movement, yet there's this tremor that seems like there's more movement. And that's further sort of complicated by the fact that, you might see Parkinson's patients who have these wild, like sort of uncontrolled movements where they're like swinging their arms around, but that is actually an over-medication phenotype where they've taken too much L-DOPA, so they're having too much dopamine production, and so it actually has the opposite phenotype. Okay, so the tremor is actually from a distinct mechanism from the slowness of movement, and it's probably a defect in the cortical, essentially the cortical waves in the motor cortex get disrupted secondarily probably to the overall motor symptoms. Okay, there is also rigidity, right, which is associated with a lack of being able to move. Postural instability, which just means that people fall a lot, right, because they lack the motor ability to stay upright. And a less known characteristic of Parkinson's is that it actually also can have pretty serious cognitive and psychological or psychiatric pathologies as well. So on the left here, I'm showing a picture of one of the more famous people who has Parkinson's. So this is Michael J. Fox. He was diagnosed when he was 30, actually. So this is a really early onset case. And it was back in 1991. So that's like 28 years ago. And so he has sort of publicly been progressing through this disease. And you can see that on his face, you can kind of see this, one of the symptoms that is associated with Parkinson's patients, which is this kind of lack of ability to emote in your face. And so it produces this kind of mask-like appearance. So coincidentally, my father-in-law, who is shown on the right here, was actually diagnosed at almost exactly the same time as Michael J. Fox, and at almost exactly the same age. And so you can see him from a picture a couple of years ago actually showing that same sort of mask-like look on his face. So I've seen firsthand how horrible this disease is. It's really bad, especially, you see a lot of patients and Parkinson's patients, they're in wheelchairs or whatever. They can't walk around very easily. But one of the really sort of tragic things about this disease is it's not inherently terminal, right? So it really progresses to be very, very severe before patients will actually die, because nothing about it is actually directly terminal. My father-in-law actually died a few months ago, and it was from aspiration or something. These patients just basically get so bad from a motor function standpoint that something else bad happens to them. So it's really horrible, and an important kind of disease to understand. Okay, so of course, Parkinson's is, pathophysiologically, as everybody knows, is associated with lack or loss of dopaminergic neurons. And so this is an image showing a slice of brain from a Parkinson's patient on the top and a normal control on the bottom, where the dopaminergic neurons are stained, and you can see that there is a prominent loss of dopaminergic neurons in this Parkinson's patient in the substantia nigra. Much like what we've talked about with Alzheimer's, where the pathophysiology is way ahead of the symptoms, you actually have to lose about 80% of your dopaminergic neurons before you exhibit any symptoms of Parkinson's. So by the time you actually are diagnosed as a patient, you are down to less than 20% of your dopaminergic neurons, because the brain is so able to compensate for various losses. Okay, so I do not expect you to remember anything from this chart. I'm just putting it up there as an illustration of a number of genes that have been associated with the onset of Parkinson's. And we're just going to talk a little bit about alpha-synuclein here, which is inherited in a dominant fashion if these particular mutations in alpha-synuclein, and it's been associated with early onset disease. But not all of these genes have been associated with early onset, of course. Some of them are associated more with late onset, and the inheritance is much less clear. So alpha-synuclein is kind of like the A beta of Parkinson's, right? It is a protein that aggregates in, and the aggregates are associated with the disease. And in this case, they form these structures called Lewy bodies. And so basically, alpha-synuclein, goes from a monomer into a protofibril, and then into a fibril state, which is amyloid-like, right? and then those then aggregate into these Lewy bodies, which are found in the brains of Parkinson's patients. So this is just an image showing some Lewy bodies, which are not entirely alpha-synuclein, but include alpha-synuclein as a major component, and you can see that they look kind of like the plaques that we saw with Alzheimer's. This is it's very complicated slide for Parkinson's. In this case, alpha-synuclein aggregation is shown at the center of it. There's a lot of other stuff involved. It seems that mitochondria are somehow important here. There's actually been some new research showing a particular mitochondrial protein that seems to have a protective function in the progression, at least of mouse models of Parkinson's. Dopamine can get oxidated. There's some oxidative stress components. It's really unclear how all of these things interact to actually produce disease. But again, the take-home point here is that it's complicated. What I do really want to talk about in more detail is how this affects movement. Okay, so hopefully, based on what we learned about in the lecture on action selection and voluntary movement, that you have kind of a general idea of how the loss of dopaminergic neurons can actually be associated with a movement disorder, right? We talked about the things like how activation of the D1 pathway, the GO pathway will initiate movement, activation of D2 will inhibit movement, how they each express different dopaminergic or dopamine receptors, right? The D1 pathway expresses D1 receptors, which actually, where dopamine increases excitability, whereas the D2 pathway is inhibited by dopamine, right? And so this is just another illustration of this whole idea, but with a focus on what happens during Parkinson's. Okay, so we have the striatum, which we talked about, right? There are these MSNs, medium spiny neurons within the striatum, and they are of these two classes, the D1 and the D2 pathways, right? So these neurons receive information from the cortex and other regions, telling the brain or the striatum things like, the sensory information that's coming in, voluntary commands of movement, internal state, like am I hungry or thirsty or whatever. This can all impinge on the striatum to drive the activation of either the D1 or D2 pathways to initiate particular movements or inhibit them or what have you, right? So it turns out that when there's lots of dopamine around, the D2 pathway favors LTD, okay? And there's very little LTP onto D2 neurons. However, D1 neurons are the opposite. So if there's lots of dopamine around, then LTP is favored, and there's actually no observable LTD on the D1 pathway. Okay, so what that means is if there's lots of dopamine, which especially happens when a normal individual has received reward in some context, right? And so there's like a bunch of dopamine dumped on there. What tends to happen then is the strengthening of the connections between the cortical inputs to the striatum and the D1 pathway. And what that does is it means that the next time those inputs are there, and so the next time you're in that situation, you will tend to initiate the same sorts of behaviors. So the example that I used was like, opening the refrigerator door and seeing a cake and eating the cake, and then the next time you're at the refrigerator, you're more likely to open the door, etc. So that's the idea when everything is working normally. If you lose a large proportion of your dopaminergic neurons from the substantia nigra, then what happens is that these preferences all flip. So now the D2 pathway favors LTP, and the D1 pathway favors LTD. All right, and so with the lack of dopamine, what that means is now all of a sudden the connections between the inputs to the striatum and the D2 pathway are being strengthened, and the connections to the D1 pathway are being weakened. And so the next time, you're in a situation where you want to initiate movement, like get up out of your chair or walk across the room or whatever it is, the pathways that actually initiate that movement have been weakened, and the pathways that inhibit the movement have been strengthened. And so you are more likely to not be able to initiate that movement. And that's why Parkinson's patients exhibit freezing behaviors, right? Where they're unable to initiate some sort of movement. Okay, so there is a learning objective here to explain the changes in the striatum in Parkinson's patients and how they relate to Parkinson's symptoms. Any questions? Yeah. Yeah, it's a good question. So I don't know a lot about this aside from anecdotal, right? And it seems to some degree that speech is much like other motor functions, that it degenerates over time, right? And so there is an increase or a decrease in the ability to initiate speech to some degree, and it sort of becomes harder and harder to understand, presumably because the fine control of like the vocal structures is degrading. But how strongly it's affected relative to other motor functions, I don't really know. Any other questions? Okay. So in the last few minutes, I just want to talk about some Parkinson's treatments. And so the frontline, like main treatment for Parkinson's patients is to just try to elevate their dopamine levels. Okay, but dopamine actually doesn't cross the blood-brain barrier. And so the drug that's given is a precursor of dopamine called L-Dopa, right? And so L-Dopa will cross the blood-brain barrier, get taken up by dopaminergic neurons, and then via an enzyme called dopa carboxylase gets converted to dopamine. And so the idea is that, maybe you've only got 15% of your dopaminergic neurons left, but if you can just increase the amount of dopamine that each one is producing, you can kind of stave off the symptoms a little bit. Okay, so this is like the #1 treatment for Parkinson's patients. It works really well for a long time, and typically, because it's able to kind of boost those levels back up close to the normal, or not normal, but enough so that you can function, right? And this is the case where I mentioned before, where if you see Parkinson's patients that are like flailing around wildly, it's because they've got too much L-Dopa, they've over-medicated. Like, this has to be controlled really, really precisely. So, there's certain numbers of pills that you have to take on basically like an hourly basis, essentially, and to really kind of try to keep those dopamine levels correct. A more aggressive treatment that is available and actually helps a lot of patients is called deep brain stimulation. And in this treatment, essentially, electrodes are implanted deep into the brain, and where they stimulate various regions, including some of the notable players in the basal ganglia circuits we've talked about before. So for example, the motor part of the thalamus is an important target of DBS, as is the GPI, the globus pallidus internal segment, which is part, if you go back in your reviewing prior to the exam of the various regions involved in that pathway, is one of the key players. So stimulation of this region affects or seems to help with the various symptoms of Parkinson's. So I'm going to show you a video of a Parkinson's patient, and the symptom that is the focus of this video is the tremor, right? Because it's one of the more obvious things that you can see, especially sort of early on in the disease. This guy is fairly young for a Parkinson's patient, so his disease probably is not super progressed. And
hey, it's me again. I just thought it would be kind of cool because I am worn off. I mean, it's been 7 hours without any kind of Sinemet, and I'm tremoring pretty good. So again, like I said, I'm a little nervous about filming the video, but I figured it'd be pretty cool if I turn my stimulator off to show you how bad I am, and turn it back on to show you how steady it keeps my hands at least. Just took my CR, so I mean I just swallowed it, so it's gonna be a little while before it kicks in, but I figured I'd turn my stimulator off and show you guys really how bad I am, and then turn it back on and show you how it smooths out. I mean, like I said, I can feel that it's off, but I can also feel that you know, it's helping me still a little bit, you know, as far as the speech and everything else goes. But I mean, I can still hold my hand, my right hand, I mean, rock steady. I mean, it's got a little tremor going there, but it's been 7 hours without medication. So here, I'm going to turn the stimulator. Let's see. Right now, as you can see, I'm set for, I don't know if the glare shows, but I'm set for 3 volts on the right side, 3.4 on the left, and I don't know if you can see it there. But I'm going to turn her off now, and we're going to shake. Again, for stimulator. Stimulator's on, 3 volts, pumping away. I'm trying to relax. Like I said, I'm a little nervous. First time I've like filmed a video like this. But anyways, let's shut her off and see where I'm at. Alright, she's definitely off. And as you can see, my legs are shaking pretty bad, and even my trunk is starting. My jaw would start too, pretty bad too, but oh, there it goes. Alright, so I mean, that's my whole body right now is tremoring. It's like a jackhammer, and as you can see, my it's even affecting my voice a little bit. So very uncomfortable, but this is pretty, I just wanted to do this. I'm going to turn her back on just to show you. This is the best part. Okay. Okay, it's good. Here we go. Okay, she's on. Takes a few. Ooh, there's the tingle. Trunk has stopped. Legs have stopped. And there's a tingle on my hands. There we go. Well, I'll show you my right hand. My right hand's the best because we still got to tweak the left hand off, but as you can see, with no Sinemet, I mean, that's night and day. I mean...
Alright, so this is something that helps a lot of patients. Obviously a much more invasive procedure. And like the administration of dopamine, it can't last forever, right? Because it still depends on the neurons being present to some degree, and ultimately you just lose too many neurons, and none of these treatments really work. Okay, so hopefully I'll see many of you on Wednesday. You've managed to find your way here, where we'll talk about the future of neuroscience.